All right, this PowerPoint presentation is on the basics of DNA and cell division. This will correlate with both the Explorations and Essentials Chapter 3. Let's talk first about the different types of cells. There are prokaryotic and eukaryotic cells. The vast majority of the semester we'll be learning about eukaryotes, since this is a class on humans. Prokaryotic organisms are single-celled organisms that do not have nuclear membranes or distinct organelles or components of the cell. Their DNA is single-stranded and contained within the cytoplasm. Prokaryotic organisms are the very first known life on Earth, and there's evidence that they originated 3.5 billion years ago. Eukaryotic cells are multi-celled organisms, so all plants and all animals. They do have a membrane-bound nucleus. The DNA is double-stranded, and there's two different types of DNA. The nuclear DNA will be contained within the nucleus of the cell, and the mitochondrial DNA will be contained within the mitochondria. Eukaryotic organisms do have specialized structures known as organelles. We'll talk about the three that will be important for this class. And evidence suggests that eukaryotic organisms originated about 1.2 billion years ago. This image here is going to show you a eukaryotic cell. The organelles that are going to be important to us, the nucleus, the purple structure in the middle, is going to contain all of the nuclear DNA. Nuclear DNA will coil around proteins called histones to make visible chromosomes. So nuclear DNA is contained within the nucleus. This is also where DNA replication occurs in the first stage of protein synthesis. The second organelle I want you guys to be aware of is this red oval shape here. This is the mitochondria. The mitochondria will house the mitochondrial DNA. It is also known as the powerhouse of the cell. It produces and stores ATP, which is basically the gasoline for the cell. And then the third organelle that will be important for this course are the ribosomes. So those are the little blue dots that you see on the endoplasmic reticulum. The ribosomes will be necessary during protein synthesis to read the messenger RNA strand. We'll talk more about protein synthesis later on in this PowerPoint. All right, so within a eukaryotic organism, there are different classes of cells. Somatic cells, also known as body cells, make up the vast majority of all the cells within our body structures, tissues, organs, nerves, bone cells, etc. All somatic cells are what we call diploid. So diploid means that they contain an entire set of chromosomes. So in a typical human will have 46 chromosomes in the nucleus of every somatic cell. Gametes are haploid, meaning that they contain half of the chromosomes. So 46 divided by 2 is 23. So every single gamete or sex cell will have 23 chromosomes in humans. So the female gamete is the ova or the egg cell. The male gamete is the sperm cell. So each and every one of us are quite literally half genetically, half mom and half dad. We receive 23 chromosomes from mom's egg and 23 chromosomes from dad's sperm. So once the sperm and egg combine, you get a zygote. So a zygote is a fertilized egg. So 23 from the sperm, 23 from the egg, you get back to the 46 total. All right. Let's talk a little bit about DNA. So the DNA molecule is essentially our template or our blueprint for all of our physical structures. It is structured as a double helix, ladder-like structure. So the rungs of the ladder here are made up of alternating complementary nitrogen base pairs. So in DNA, adenine will always pair with thymine, and cytosine will always pair with guanine. So we'll review that a couple more times. It'll be important. A always pairs with T, C always pairs with G. It'll be a little bit different when we start learning about RNA, but to avoid confusion, let's focus on DNA first. So always adenine to thymine and cytosine to guanine. All right, so DNA is made up of nucleic acids. The building blocks of the nucleic acids are called nucleotides. So each nucleotide will have three main components. The first being the sugar molecule. So if you're looking at DNA, DNA has a type of sugar called deoxyribose. That's where you get the D in DNA. Whereas RNA will have a sugar called ribose, which is where you get the R in RNA. The second component is the phosphate molecule, and then the rungs of the ladder are made up of the alternating complementary nitrogen base pairs. So again, adenine always pairing with thymine, and cytosine always pairing with guanine, and vice versa. All right, the two different types of DNA, there's nuclear DNA and mitochondrial DNA. Nuclear DNA is the actual genetic material that makes up our chromosomes. So nuclear DNA will coil around proteins called histones to form visible chromosomes. So in every single somatic cell of a typical human, 46 individual chromosomes. A complete set of all 46 is called a genome. Nuclear DNA is homoplasmic, which refers to nuclear DNA which is identical in the nucleus of each cell type. But of course, cells will differentiate. Some cells will specialize to become skin cells, some become organ cells, some become blood cells. You guys get the picture. All right, so that's what nuclear DNA looks like underneath a microscope. All right, and then the other type of DNA is called mitochondrial DNA. You might see it abbreviated as mtDNA. So mitochondrial DNA is unique because it's inherited exclusively from the mother. So if you're reading a DNA study and they're interested in tracing down the matriline, or mother to grandmother to great-grandmother, so on and so forth, the matriline can be traced back hundreds of thousands of years, and that search for that mitochondrial Eve, or it's a way of tracing your maternal lineage. All right, DNA is going to be responsible for two vital processes that we'll learn about here in this PowerPoint presentation. The first being DNA replication, and the second being protein synthesis. So DNA replication is going to occur entirely in the nucleus of the cell, and it occurs during the interphase or prep phase portion for both mitosis and meiosis. So mitosis will be the creation of somatic cells, whereas meiosis is the creation of gametes or sex cells. So the basics of what happens during DNA replication, there's an enzyme called DNA helicase that will inspire the parent strand to unzip. So in this image here, the blue ribbon represents the parent strand or the original strand, and then the gold ribbon is representing the daughter or the new strand. So DNA helicase inspires the parent strand to unzip, which exposes all of those nitrogen bases, so all the A's, T's, C's, and G's. And then the daughter strand will come in and pair up, following those rules of adenine always pairing with thymine and cytosine always pairing with guanine. So your end result is two identical strands of DNA. The reason this is important is during cell division, we must have the new cell. Once we get to the conclusion of cell division, the new cell is also going to need an entire set of the genome, all 46 chromosomes. So that's why DNA replication must occur during interphase to prepare for cell division. All right, so here's where the differences come in. So in DNA, we've got adenine pairing with thymine, guanine pairing with cytosine, thymine pairing with adenine, cytosine pairing with guanine. In RNA, the only difference is adenine will now pair with a new nitrogen base called uracil. So that'll be important when we talk about protein synthesis. So only difference, RNA, adenine is now going to pair with uracil. All right, let's talk about protein synthesis. So proteins are the chemicals that make up our tissues. They also regulate functions, repair, and they are responsible for the growth of our bodily tissues. Proteins are made up of amino acids. We have 20 different types of amino acids that will combine in various sequences to make specific proteins. So amino acids are organic molecules that combine in a specific sequence read by the ribosomes to form proteins. And essential amino acids are proteins that form an organism's physical attributes. Some of these essential amino acids we must get from our diet. All right, different types of proteins. There's structural proteins and regulatory proteins. Structural proteins are responsible for our physical characteristics. So for example, keratin and collagen are forming the structure in our skin and nails. And regulatory proteins are responsible for bodily functions. So some examples of that would be enzymes, hormones, and antibodies. Protein synthesis will involve two main steps. So we'll talk about it in more detail here in a moment, but the two steps are transcription and Translation. All right, so the first step, also called transcription, is going to occur entirely in the nucleus of the cell where the nuclear DNA resides. So it will involve the DNA template coding for the messenger RNA strand. So messenger RNA is abbreviated with a lowercase m and then RNA. Be careful not to mix that up with mtDNA. mRNA is small enough to leave the nucleus and travel out into the cytoplasm where the ribosomes are located. So during the second step, also known as translation, the messenger RNA will carry the message into the cytoplasm and essentially translate codon by codon into a specific order of amino acids, or a polypeptide chain. So DNA is too large to leave the nucleus, which is why we need RNA for the conclusion of protein synthesis, because RNA is small enough to leave the nucleus, travel into the cytoplasm where the ribosomes are located. The ribosomes are necessary to read or translate the code. All right, so here are the two steps right next to each other. So transcription is occurring entirely in the nucleus of the cell. It results in a complementary copy or blueprint of DNA. And then from this DNA template, that's how the messenger RNA strand is produced. So the messenger RNA strand will leave the nucleus and travel out into the cytoplasm. So then once we're out into the cytoplasm, we're into phase 2 of protein synthesis. That's called translation. So translation will occur entirely in the cytoplasm where the ribosomes are located. The messenger RNA carries its message of the sequence of DNA bases, and the ribosomes will essentially read the messenger RNA bases 3 at a time. So AUG, for example, is the start codon. So it's going to read that strand codon by codon. The transfer RNA will then bring in the appropriate amino acid into place. And the messenger RNA strand now has formed the polypeptide chain. So you break that word up. Poly means many, peptide means protein. So this chain of amino acids is going to code for a specific protein. All right, so here it is broken down into a chart format here. So you're starting with your DNA sequence, which then codes for the messenger RNA sequence. So those first two steps are occurring in the nucleus. Then the messenger RNA strand will leave the nucleus, travel out into the cytoplasm where the ribosomes are located. The ribosomes read the code, codon by codon, or 3 by 3. And then you will have an amino acid sequence, also called a polypeptide chain. And then your end result is a specific protein. So the proteins code for the genes on our chromosomes. All right, let's talk about the different types of chromosomes. There's 2 types of chromosomes in humans. So pair number 1 through 22, those are called autosomes. And then your 23rd pair would be your sex chromosomes. So if you are biologically female, then you carry only X chromosomes. You are XX on your 23rd pair. If you are biologically male, you have 1 X chromosome and 1 Y chromosome. Now, there are actually some chromosomal aberrations and abnormalities that result in extra sex chromosomes or missing sex chromosomes, but we'll talk about that later on here in this PowerPoint. For right now, let's focus on the standard condition. So this is an image of what a karyotype looks like. So a karyotype is basically a picture of your genome. So it's all 46 chromosomes or 23 pairs. So this picture here is a picture of a female, a biological female, because you see pair number 1 through 22. Those are her autosomes, and then she has 2 X chromosomes. So this is a biological female. And you also notice that the chromosomes are organized by size and centromere location. The different colorations, they stain the chromosomes using a process called gel electrophoresis, which allows you to identify the homologous chromosomes. So the different colors are representing different genes that are located on those chromosomes. So number 1 is the largest chromosome, number 22 is the smallest. So there are some chromosomal aberrations that will result in extra or missing chromosomes. For example, if there's a non-disjunction at the 21st chromosome, if you end up with 3 21st chromosomes, we call that trisomy 21, also known as Down syndrome. So here's another karyotype of a typical male, so pair number 1 through 22. Those are the autosomes, and then the 23rd pair, those are his sex chromosomes. He has one X and one Y. So this is a karyotype of a biologically typical male. All right, let's go ahead and pause here, and then in part 2, we'll go over cell division, mitosis, and meiosis.